The BCL2 protein family plays a crucial role in regulating apoptosis, tumorigenesis, and cellular responses to anti-cancer therapy. These proteins have either pro-apoptotic or anti-apoptotic activities, controlling the point of no return for cell survival. Understanding how these family members interact during programmed cell death is important, as it affects tumor development, host pathogen interactions, and animal development. Recent structural, phylogenetic, and biological analyses suggest we need to reconsider our understanding of how these proteins interact and activate caspases, the enzymes that execute cell death. While we have gained insights into the regulation mechanisms of these interactions, a unifying hypothesis for their activation of caspases remains elusive. The discovery of the BCL2 gene at the T14/18 chromosome translocation in B-cell follicular lymphomas was a significant breakthrough. Unlike previously discovered oncogenes that promote cell proliferation, BCL2 inhibits cell death, introducing a new paradigm for carcinogenesis. Apoptosis, or programmed cell death, is now widely accepted as a prominent tumor suppression mechanism. Mutations in certain oncogenes often require a second mutation to inhibit apoptosis for efficient tumor promotion. This shift in understanding the role of apoptosis in cancer development has had a profound impact on our approach to cancer research and treatment. BCL2 and MYC are two key oncogenes that work together to drive cancer development. BCL2 overexpression inhibits apoptosis, allowing cells with genetic damage to survive and creating an environment conducive for cancer. MYC overexpression drives excessive cell proliferation, increases metabolic activity, and promotes genetic instability. The combined effect of BCL2 and MYC overexpression results in potent synergy, accelerating tumor growth and increasing resistance to therapy. This synergistic relationship between BCL2 and MYC is particularly pronounced in the development of lymphomas. The BCL2 family is a critical regulator of apoptosis, or programmed cell death. It includes both anti-apoptotic members like BCL2, BCLXL, and MCL1, as well as pro-apoptotic members like BAX and BAC. BH3-only proteins like BAD, BID, and PUMA interact with and regulate the core BCL2 family to promote apoptosis. Despite their opposing functions, the BCL2 family members share remarkable 3D structural similarity. Understanding the structural and functional differences within this family is key to unraveling the complex mechanisms of apoptosis regulation. The BH3 motif is a critical functional domain found in many BCL2 family proteins. This short amino acid sequence, LXXXGD, is highly conserved and plays a key role in the structure and function of these proteins. The BH3 domain forms part of the hydrophobic pocket in core BCL2 family members, which is crucial for their protein-protein interactions. In BH3-only proteins, the BH3 domain mediates their interaction with anti-apoptotic BCL2 family members, either inhibiting them or directly activating pro-apoptotic proteins to promote cell death. Understanding the sequence, structure, and function of the BH3 motif is critical for understanding the complex regulation of apoptosis by the BCL2 family. The slide provides an overview of the two main pathways that can lead to apoptosis, or programmed cell death: the intrinsic and extrinsic pathways. The intrinsic pathway is triggered by internal cellular stresses like DNA damage or growth factor deprivation, and is regulated by the BCL2 family of proteins, leading to activation of caspase 9. The extrinsic pathway is triggered by external signals, specifically the binding of death receptors like FAS or TNFR1, which recruit and activate caspase 8 through the FADD adapter protein. Both pathways ultimately converge on the activation of caspases, which coordinate the dismantling and removal of the doomed cell. It's important to understand the distinction between these two pathways, as they can be triggered by different stimuli and involve different regulatory mechanisms. The slide outlines the intersection between the extrinsic and intrinsic apoptosis pathways, focusing on the role of the BID protein. It shows how activation of death receptors like FAS or TNFR1 leads to caspase 8 activation, which then cleaves BID to form the truncated TBID. TBID then translocates to the mitochondria, where it promotes further caspases activation through the intrinsic apoptosis pathway. This connection between the extrinsic and intrinsic pathways is particularly important in certain cell types like hepatocytes. The slide uses a detailed molecular illustration to visually explain this pathway intersection and the role of BID. The BCL2 family of proteins play a key role in regulating apoptosis, or programmed cell death. There are three main classes of BCL2 family members with opposing functions. The anti-apoptotic members like BCL2 and BCLXL inhibit the pro-apoptotic proteins, BAX and BAC, to prevent cell death. In contrast, the pro-apoptotic members BAX and BAC induce permeabilization of the mitochondrial outer membrane, leading to the release of apoptogenic molecules and caspases activation. The BH3-only proteins, like BID and BIM, act as sensors and can bind and inhibit the anti-apoptotic members, thereby repressing BAX and BAC to trigger apoptosis. Understanding the delicate balance between these opposing activities within the BCL2 family is crucial for regulating cell survival and death. Mitochondria are the powerhouses of the cell, providing energy through cellular respiration. The outer membrane of mitochondria plays a crucial role in apoptosis, or programmed cell death. Two key proteins, BAX and BAC, undergo changes that allow them to form pores in the outer mitochondrial membrane. These pores enable the release of proteins like cytochrome C from the space between the inner and outer membranes. The release of these proteins triggers a cascade of caspases activation, leading to apoptosis. This process of mitochondrial outer membrane permeabilization, MOMP, is a critical step in the intrinsic apoptosis pathway. Understanding MOMP is important for understanding how cells regulate and undergo programmed cell death. The apoptosome is a crucial protein complex that activates the caspases cascade, leading to programmed cell death or apoptosis. It is formed when cytochrome C is released from mitochondria and binds to the APAF1 protein, causing it to assemble into a heptameric ring structure. This apoptosome structure can then bind to and activate the pro-caspases 9 enzyme, setting off a chain reaction of caspases activation that demolishes the cell. While this cytochrome CAPAF1 caspases 9 pathway is critical for apoptosis in many cell types, lymphocytes can use alternative caspases activation mechanisms that are independent of this pathway. The slide introduces the topic of inhibitor of apoptosis proteins, IAPs, and their role in regulating cell death. IAPs like XIAP bind and inhibit certain caspases enzymes, preventing apoptosis or programmed cell death. Diablo, also known as SMAC, is a protein released from mitochondria that can antagonize IAPs, relieving their inhibition of caspases and allowing cell death to proceed. Surprisingly, mice lacking either Diablo or XIAP don't show significant apoptotic phenotypes, suggesting there are additional, redundant pathways for caspases activation that are yet to be discovered. The slide is accompanied by a molecular illustration showing the interaction between XIAP, caspases 9, and Diablo. The BCL2 family proteins appear to control cell survival beyond just caspases activation. Even when caspases activation is blocked, cells can still lose their ability to divide and effectively die. The step regulated by the BCL2 family seems to be a critical final commitment point in the decision between cell life and death. Disruption of the mitochondria by BAX and BAC may ultimately lead to cell death, even without activating the apoptosome. When caspases are blocked, cells may die more slowly through BCL2 family-mediated mitochondrial disruption or novel caspases activation pathways. The slide introduces the topic of evolutionary conservation, focusing on the apoptosis pathway in the model organism C. elegans. It outlines the key players in the C. elegans apoptosis pathway: EGL1, CED9, CED4, and CED3. EGL1 is a BH3-only protein that is induced by developmental cues and binds to CED9, a BCL2 homologue. This frees CED4, an APAF1-related protein, to assemble into a tetrameric apoptosome and activate the caspases CED3. This model differs from the cytochrome C release model in mammals, suggesting there may be underlying common processes among BCL2 family members across species that are yet to be discovered. The goal is to highlight the simpler apoptosis pathway in C. elegans and how it can provide insights into more complex pathways in other organisms. The BCL2 family of proteins play a crucial role in regulating apoptosis, or programmed cell death. Despite their opposing functions, the core structural features of pro- and anti-apoptotic BCL2 family members are remarkably similar. Anti-apoptotic proteins like BCL-XL have a helical bundle structure with a hydrophobic helix-turn helix hairpin flanked by amphipathic helices. Pro-apoptotic proteins like BAX share this same basic fold, with no obvious structural differences to explain their opposing functions. The BH3-only protein BID is a structural outlier, with a 3D structure more similar to the core BCL2 family members, allowing it to oligomerize and permeabilize membranes. Understanding these structural similarities and differences provides insight into how the BCL2 family regulates the delicate balance between cell survival and cell death. The slide discusses how some viruses have evolved proteins that mimic the structure of BCL2 family proteins, even without sequence similarity. This highlights the importance of the BCL2 structural fold in regulating apoptosis, or programmed cell death. The slide provides examples of three viral proteins case BCL2, M11L, and N1L that adopt a similar helical structure to BCL2 family members, allowing them to inhibit apoptosis and counteract host defense mechanisms. Despite differences in sequence, these viral proteins are able to mimic the function of BCL2 proteins through structural mimicry, demonstrating the critical role of this structural fold in regulating this key cellular process. The BCL2 family of proteins are central regulators of apoptosis, or programmed cell death. The core BCL2 family members share homology across multiple BCL2 homology domains and have similar predicted secondary structures, indicating they are evolutionarily related. This core group includes BCL2, BCLXL, BCLW, BAX, BAC, BOC, MCL1, A1, and BCLB. BID is an exception. It has a structure similar to the core family, but is the only BH3-only protein that shares this evolutionary relationship. BID has unique properties like the ability to oligomerize and permeabilize membranes, similar to BAX and BAC. The other BH3-only proteins, like BAD, BMF, and BIM, have unrelated structures to the core family. These BH3-only proteins acquired their BH3 motifs through convergent evolution, and many are intrinsically unstructured. They share only the BH3 motif for binding to the core BCL2 family members. Mule is an E3 ligase, a type of enzyme that attaches ubiquitin to other proteins, targeting them for degradation. Mule contains several key domains: UBA domain that binds ubiquitin, WWE domain that facilitates protein-protein interactions, HECT domain that carries out the ubiquitination process, BH3 domain, which is typically found in pro-apoptotic BCL2 family proteins. The BH3 domain allows Mule to interact with and regulate other BCL2 family members, even though Mule itself is not a core BCL2 protein. Mule's main function is to ubiquitinate and degrade the anti-apoptotic protein MCL1, thereby promoting cell death. This adds an additional layer of control over the apoptotic machinery, beyond just the canonical BCL2 family members. BH3-only proteins are key initiators of apoptosis, or programmed cell death. They can be activated through various mechanisms, allowing cells to respond to diverse death signals. One way is through transcriptional induction. P53 induces NOXA and PUMA after DNA damage, while FOXO3A induces BIM after growth factor deprivation. Proteolytic activation is another mechanism, where BID is cleaved by caspases 8 during death receptor signaling. Phosphorylation changes can also activate BH3-only proteins. For example, loss of phosphorylation activates BAD, and loss of ERK-mediated phosphorylation activates BIM. Finally, BH3-only proteins can be released from sequestration: BIM from the dynein motor complex, and BMF from actin-myosin motor complexes. The ability of BH3-only proteins to be activated through these diverse mechanisms allows cells to engage the core apoptotic machinery in response to a wide range of death stimuli. The slide outlines the key mechanisms that regulate the expression and stability of anti-apoptotic BCL2 family proteins, which play a critical role in controlling cell survival and death. Transcriptional regulation. BCL-XL can be induced by growth factors through the JAK-STAT pathway, promoting cell survival. Protein stability. MCL1 is rapidly degraded by the proteasome in response to death stimuli like cytokine deprivation or UV radiation. Post-transcriptional upregulation. MCL1 levels can be increased post-transcriptionally, preventing apoptosis by inhibiting its degradation. These regulatory mechanisms allow cells to quickly adjust the balance between pro-survival and pro-death signals, providing an important control point for apoptosis. The table shows the binding specificity of different BH3-only proteins to anti-apoptotic BCL2 family members. BIM and PUMA bind to all 5 anti-apoptotic proteins tested, indicating their potent pro-apoptotic activity. TBID shows weaker binding to BCL2, but strong binding to the other 4 proteins. BAD binds selectively to BCL2, BCLXL, and BCLW, but not MCL1 or A1. NOXA only binds to MCL1 and A1, suggesting a more specific role in regulating these 2 proteins. The different binding profiles of these BH3-only proteins allow for fine-tuned regulation of apoptosis in different tissues and contexts. The BCL2 family of proteins play a crucial role in regulating apoptosis, or programmed cell death. The anti-apoptotic members like BCL2, BCLXL, BCLW, and MCL1 are located in different cellular membranes, including the endoplasmic reticulum, nuclear envelope, and outer mitochondrial membrane. During apoptosis, the pro-apoptotic members like BAX and BAC undergo conformational changes and translocate to the mitochondria. BAX is mostly found in the cytosol in healthy cells, but it inserts into the outer mitochondrial membrane during apoptosis. BAK, on the other hand, is already located on the outer mitochondrial membrane, where it is normally bound to anti-apoptotic proteins like MCL1 and BCLXL. These interactions are disrupted during apoptosis. The subcellular localization of these BCL2 family proteins is critical for their pro- or anti-apoptotic functions. The slide outlines the key conformational changes that occur in the BAX protein during the process of apoptosis, or programmed cell death. In healthy cells, BAX exists as a soluble, folded protein in the cytoplasm. Its C-terminal helix and amphipathic helices are tucked within the protein structure. During early apoptosis, the N-terminus of BAX, amino acids 1327, becomes exposed and accessible to antibody binding, indicating a significant structural change. As apoptosis progresses, BAX undergoes extensive remodeling, with at least 3 of its helices inserting deeply into the mitochondrial membrane. This allows the protein to oligomerize and form pores. Both pro-apoptotic and anti-apoptotic BCL2 family members, like BAX, go through dramatic conformational changes during apoptosis, including increased proteolytic sensitivity and oligomer formation as BAX translocates to the mitochondria. Apoptosis, or programmed cell death, is a crucial process in our bodies. 2 key proteins involved in this process are BAX and BAC. These proteins undergo distinct steps of activation during apoptosis. First, they translocate to the mitochondria and undergo a conformational change in their N-terminal region. This exposed N-terminal epitope can be detected using a specialized monoclonal antibody. The oligomers formed by BAX and BAC during this process have a wide range of molecular weights. The exact number of BAX and BAC units in these oligomers is still unclear and an active area of research. Anti-apoptotic BCL2 proteins undergo conformational changes during apoptosis when they bind to BH3-only proteins. This causes BCL2 to insert into the membrane, resembling the structure of pro-apoptotic BAX. However, unlike BAX, BCL2 does not proceed to oligomerize after membrane insertion. Instead, BCL2 and BCLXL may bind to BAX and BAC after they insert into membranes, capping the oligomers and inhibiting their chain elongation. This capping function is a key way that anti-apoptotic BCL2 proteins can inhibit apoptosis. In healthy cells, the conformation of BCL2 does not allow it to bind to pro-apoptotic BAX. But during apoptosis, the conformational changes enable BCL2 to interact with and inhibit activated BAX and BAC. The release of cytochrome C from mitochondria is a critical point of no return in the apoptosis process. This begins with the activation and oligomerization of pro-apoptotic BCL2 proteins like BAX and BAC at the outer mitochondrial membrane. This leads to permeabilization of the outer mitochondrial membrane, allowing soluble intermembrane space proteins to be released into the cytosol. Proteins like cytochrome C, Diablo, AIF, and others are released and activate caspases, the executioners of apoptosis. This irreversible caspases activation leads to the systematic demolition of the cell. Understanding this cytochrome C release step is crucial, as it represents the point of no return in the apoptosis pathway. The structure of BCL-XL, a pro-survival BCL2 family member, is remarkably similar to the translocation domain of diphtheria toxin, suggesting it may have a pore-forming function. Interestingly, both pro-apoptotic (BAX) and anti-apoptotic (BCL-XL) BCL2 family members can form membrane channels in vitro, which is confusing. Functional evidence shows that BAX can induce the release of cytochrome C from isolated mitochondria, and allow the release of large dextrin molecules from liposomes, consistent with the formation of lipidic pores. BCL-XL is able to inhibit both BAX-induced cytochrome C release from mitochondria and dextrin release from liposomes, suggesting it may regulate these pores. However, key questions remain unanswered, such as the biochemical nature of the BAX-BAC pore, the number of BAX molecules that comprise the pore, and how anti-apoptotic BCL2 family proteins like BCL-XL regulate these pores. Neurons and certain other cell types can survive for a time even after releasing cytochrome C, a key step in apoptosis. This is due to tight regulation of caspases by inhibitor of apoptosis proteins, IAPs. Cardiomyocytes also show resistance, requiring additional signals like Diablo release to overcome IAP inhibition. This specialized adaptation may provide extra protection for long-lived, essential post-mitotic cells that cannot be easily replaced. The additional regulatory layer helps prevent accidental cell death in these critical cell types. The slide discusses how mitochondria undergo fragmentation during the process of apoptosis, or programmed cell death. It highlights three key events that occur. BAX and BAC proteins form small focal regions on the mitochondrial surface, which is an early sign of mitochondrial activation. These BAX foci then lead to mitochondrial division and fragmentation, which happens almost simultaneously with the release of cytochrome C from the mitochondria. The mitochondrial fission protein DRP1 is involved in this fragmentation process, and inhibiting DRP1 can delay cytochrome C release and caspases activation, suggesting the fission machinery plays a role in regulating apoptosis. The images provide visual examples of these mitochondrial changes during apoptosis induction. Healthy cells lacking both BAX and BAC have altered mitochondrial morphology and slower mitochondrial fusion rates. BAX and BAC affect mitochondrial morphogenesis machinery even in the absence of apoptotic stimuli. OPA1 controls mitochondrial cristae formation, and tight cristae junctions can inhibit cytochrome C release during apoptosis. DRP1 knockdown inhibits cytochrome C release but not the release of other proteins like Diablo. The connection between mitochondrial morphology and apoptosis regulation is an important area of ongoing research. BAX and BAC are two pro-apoptotic proteins that play a key role in regulating cell death. Mice lacking just one of these proteins, either BAX or BAC, have relatively mild phenotypes. They are viable and mostly normal. However, mice lacking both BAX and BAC have severe defects and often die during development or shortly after birth. This dramatic difference between the single and double knockout mice demonstrates the extensive redundancy and overlapping functions of BAX and BAC. Cells lacking both BAX and BAC are highly resistant to most forms of stress-induced apoptosis, confirming that these two proteins are essential downstream effectors of the cell death pathway. The redundancy between BAX and BAC highlights their critical importance in controlling programmed cell death. The BH3-only proteins play a crucial role in regulating apoptosis, or programmed cell death. Gene knockout studies have revealed the specific functions of different BH3-only proteins. Loss of the BH3-only protein BIM leads to abnormal accumulation of lymphoid and myeloid cells, and can cause a fatal autoimmune disease similar to systemic lupus erythematosus, SLE. BIM is essential for deleting autoreactive T and B cells, and for inducing apoptosis in response to growth factor deprivation. BID-deficient mice are resistant to FAS-induced apoptosis in hepatocytes. Puma-deficient cells show resistance to DNA damage-induced apoptosis. Other BH3-only proteins like BAD, BIC, HRK, BMF, and NOXA have milder phenotypes when knocked out, reflecting their more limited binding to anti-apoptotic proteins. BIM and PUMA are the most crucial BH3-only proteins for initiating apoptosis, or programmed cell death. They can bind to and inhibit all 5 major anti-apoptotic BCL2 family members. Losing both BIM and PUMA provides as much protection against cell death as overexpressing the anti-apoptotic protein BCL2 or deleting the pro-apoptotic proteins BAX and BAC. Analysis of mice lacking both BIM and PUMA has shown they are the most important for apoptosis in response to many death stimuli, especially in blood and immune cells. While losing just BIM or PUMA alone makes cells resistant to certain death triggers, only losing both provides the maximum protection against cell death. In terms of BIM and BCL2, a critical balance. It discusses how the loss of a single BIM allele can prevent the fatal polycystic kidney disease and lymphopenia caused by the loss of BCL2 in mice. Specifically, when BCL2 is absent in certain cell types, the normal levels of BIM are not sufficiently opposed, leading to abnormal apoptosis. The loss of both BIM alleles can completely prevent all the defects, including premature graying, caused by the lack of BCL2. This provides important mechanistic insight into how the levels of BIM must be balanced by BCL2 in specific tissues. The key takeaway is the interplay between BIM and BCL2 is critical for maintaining proper cell survival and function. Despite intensive research, there are still several mysteries surrounding how these proteins are regulated. One key unknown is the molecular trigger that causes the pro-apoptotic proteins BAX and BAC to become activated and induce cell death. Another mystery is what structural differences define the anti-apoptotic versus pro-apoptotic members of the BCL2 family. We also don't fully understand the functional effects of the BH3-only proteins binding to the anti-apoptotic BCL2 proteins. Difficulties in studying the structure and interactions of these proteins, especially when they undergo conformational changes or insert into membranes, have impeded progress in resolving these remaining mysteries. The slide provides an overview of the evolutionary conservation of apoptosis regulation across different species. It shows the number of core BCL2 family members and BH3-only proteins found in various organisms, from sponges to humans, mice. Sponges have 2 BCL2-related genes that resemble mammalian BOC, suggesting these genes evolved with multicellular life. C. elegans has 1 core BCL2 member, CED9, and 2 BH3-only proteins, EGL1, CED13, with EGL1 being crucial for most programmed cell death. D. melanogaster has 2 core BCL2 members, required for stress-induced but not developmental apoptosis. Sea urchins have significantly more BCL2 family members, 10, compared to insects and worms. Zebrafish and humans, mice have a similar set of 9 and 1,2,1,3 B, C, L, 2 family members, respectively, and 8 BH3-only proteins, indicating a stable organization among mammals. The evolutionary conservation of these apoptosis regulators highlights their importance in multicellular life. The BCL2 family of proteins plays a critical role in regulating apoptosis, or programmed cell death, in our cells. Viruses have evolved various strategies to exploit this regulatory mechanism and prevent apoptosis of infected cells, allowing the virus to replicate and spread. I'll highlight 3 examples of viral proteins that mimic BCL2 family members to achieve this. Case BCL2 from Kaposi sarcoma virus directly mimics the structure and function of human BCL2 to prevent apoptosis. M11L from myxoma virus adopts a BCL2-like fold despite lacking sequence similarity, allowing it to bind and inhibit BH3-only proteins. N1L from vaccinia virus also has a BCL2-like structure, demonstrating convergent evolution towards this critical regulatory mechanism. These examples illustrate the importance of the BCL2 family in cell survival and how viruses have evolved ingenious ways to subvert this pathway for their own benefit. The BCL2 family of proteins play a crucial role in regulating cell death and survival. Understanding how these proteins work has led to the development of targeted therapies called BH3 mimetics. These small molecule inhibitors can bind to anti-apoptotic BCL2 proteins, inducing apoptosis in cancer cells. Modulating BCL2 family proteins also has therapeutic potential for other diseases. In neurodegenerative conditions like Alzheimer's and Parkinson's, inhibiting inappropriate neuronal apoptosis could be beneficial. Targeting viral BCL2 homologs could prevent viruses from inhibiting host cell apoptosis, offering new antiviral strategies. In ischemic diseases like heart attack and stroke, preventing apoptosis by modulating BCL2 proteins could limit tissue damage and improve outcomes. The slide highlights key future directions in BCL2 family research, which is critical for understanding apoptosis and cell death mechanisms. Structural characterization of BACs and BAC conformational changes, oligomerization, and membrane insertion will shed light on how they form pores to induce cell death. Identifying the precise molecular triggers that activate BAC's translocation and BAC during apoptosis will reveal the upstream signaling events. Defining the structural and functional differences between pro- and anti-apoptotic BCL2 proteins will clarify their opposing roles in regulating cell fate. Discovering novel APAF1 independent pathways of caspases activation regulated by BCL2 proteins could uncover alternative mechanisms of apoptosis induction. Recent advances in understanding BCL2 family interactions, supported by animal studies and cell biology, provide a strong foundation for deciphering the remaining mysteries of cellular commitment to apoptosis.